Dr. Nurul Samshina. So she is going to talk about the effect of biofertilizers on the plant rhizosphere microbiota. So a little bit about Dr. Nurul. So Dr. Nurul Samshina is currently a senior lecturer and a plant microbiologist at ISP Faculty of Science, So she obtained her bachelor degree in agriculture science from UPM in 2009 and her PhD from in 2017. So her research interest is on plant disease diagnostic and also the soil microbiome. So without further ado, I would like to invite Dr. Nurul Samshina to share her research with us. And Dr. Nurul, the screen is yours.
Assalamualaikum warahmatullah. Very good morning to everyone. Thank you for joining me and others today for this SEBA expert seminar. So let me share my screen first. Okay. So yeah, I did quickly check the participants. I can't see people from ISP yet because now we are having our department meeting, and then later on we'll be having Jamuan Raya around 11, I guess, like almost to the Jamuan Raya. So never mind, not a very good timing, but I hope some of my collaborators will be joining this session soon. Okay, so thank you very much to SEBA for this opportunity to share a little bit of my knowledge, of course, interest and humble experience on this presented topic. So today I am going to share some of my quick reviews on this topic and tidbits from my current research related to this topic, the effects of biofertilizers on the plant rhizosphere microbiota. Okay, and thank you very much for the introduction just now. So about this topic, first I just want to introduce to some issues and factors related to rhizosphere microbiota. The environmental issues such as water shortages, destruction of biodiversity, a decline in soil fertility, and elevated levels of greenhouse gases leads to increase in biotic and abiotic stresses, right? So, which include living organisms, biotic and abiotic stresses, meaning those non-living factors that threaten agricultural productivity and food security. So this alarming situation actually rooted in the overuse of chemical fertilizers and pesticides, and I would say like over-reliance on nitrogen fertilizers, which led to soil nutrient imbalance, gradual decline in crop growth, reduction in soil organic matter, reduction in the activity of soil organisms, and all these factors actually related to plant growth, right? And we know that soil is a very complex ecosystem in which different microorganisms play different roles in towards promoting plant growth and yield, right? So the rhizosphere is the region of soil influenced by plant roots through rhizodeposition of root exudates, and these root exudates are the key determinants of rhizosphere microbiome structure. So plants and soil microorganisms actually communicating together, and then somehow leads to the or promote the plant growth and that kind of thing. And plants have been placed in a sea of microorganisms from the time where they are planted. So these soil microbes are sensitive to environmental stresses, and they play an important role in fertilizer nutrient conversion. So we should know that these soil microbes are like totally affected by those environmental stresses. And if they are stressed, and then it will also affect the fertilizer nutrient conversion for the use of plants. Okay, so talking about this environmental issues, soil, plants, so all these components actually leads us to look at the bigger picture of plant and soil ecosystem, the part of the microbiomes. Okay, so what is phytobiomes? It is a plants, their environment, and the complex world of organisms associated with them. So everything that related communicating with plants are actually part of their life cycle, I would say. So looking at this cycle or diagram here, so plants, of course, part of phytobiome, and then the climate, the environment, animals, meaning that any small animals like insects and those that can cause disease and affect the yield and productivity of crops, also part of phytobiomes. And then all the data that related to plant growth, plant yield, nutrients, of course, that relate with soil. So meaning that nutrients that are available in the soil and whatever nutrients that are being supplied for the plant growth. And of course, microbes, the soil microbes, the microbes that surrounding the plants in phyllosphere, endosphere. So they are all the microorganisms that relate to plants. Okay, so the phytobiome roadmap, a new vision for agriculture, in which sustainable crop productivity is achieved through a system-level understanding of diverse interacting components. So it is a huge term, right? Looking at all the components inside phytobiomes. This name or concept actually created in around 2013 during a meeting hosted by American Phytopathological Society. So the scientists discussed about global food crisis and the increase in human population. So they So in order to optimize crop productivity, we can no longer focus on just one or two factors that affect the crop productivity, but we have to look at all the factors that, which are interrelated under the phytobiome. So we have to explore the phytobiome. Okay, but of course it is a huge topic and involves a lot of components or factors inside, right? But just a quick probing the phytobiome to advance agriculture. So when a plant is sitting in the field, it's not just exposed to one pathogen, one temperature, one insect at that specific moment, right? It is exposed to everything at once. If you want to understand how plants respond to a single pathogen, we really need to take the whole system, which is the phytobiome, right, in order to understand the process that occur, the disease and whatnot. So scientists and farmers actually long know that many interacting factors affect crop health, but it's just that due to limiting factors, so we normally focus on one specific pathogen when there's an outbreak of disease. And the way we report diseases also, we normally trying to identify the specific causative agents, and then we need to test in laboratory conditions or glasshouse conditions. Then we can prove that that specific pathogens actually cause the disease. But then we are not looking at the whole system, right? Where there could be environmental stresses and the pathogen actually an opportunistic pathogen that somehow become virulence because of other factors that are affecting the pathogens. Okay, so with genomic tools, we can begin to put these pieces together in ways that we couldn't do 10 to 20 years ago. So now people are talking about omics, bioinformatics, and all these different omics tools in order to understand this bigger picture of phytobiomes. Okay. So today I'm going to focus on just one part, like I mentioned just now, the microbes. And even when we talk about the microorganisms, so like I mentioned, there are phyllosphere, endosphere, rhizosphere, but then today we're just going to look at the rhizospheric microbes. Okay, so impacts of biofertilizers on sustainable agriculture. So talking about rhizospheric microbes, there are many factors that affect the presence, the abundance, the diversity of those microbes. And now people are looking at the impacts of biofertilizers. As we all know, sustainable agriculture mean to improve or enhance food production, right? And then in order to have a better food, safe food, and then of course to maintain soil fertility, soil productivity, soil quality. So there's growing demand for biological-based organic fertilizers as an alternative to agrochemicals, right? So we used to understand that these biological-based organic fertilizers or biofertilizers are 100% safe, right? And then can just be applied anytime, in any amount, that kind of thing. But then now, like, people are trying to understand the impacts of biofertilizers towards those native microorganisms in the soil. Okay. So the use of biofertilizers, of course, improves soil fertility and plant performance, sustainable strategy to improve soil biodiversity, plant nutritional status and growth. So these are the things that are being understood, right? And fertilizers in microbial communities that determine fertilizer efficiency are key to sustainable agriculture development. So by applying fertilizers, actually, we also affect the microbial communities and vice versa. Okay, so let's quickly have a look at what is biofertilizer and the effects. So it has been introduced to define a fertilizer that contains plant growth promoting microbes, the acronym PGPMs, which are able to colonize plant roots and provide benefits to their hosts. So they are those microbes, bacteria, fungi, PGPB, PGPF that promotes plant growth and productivity, and of course towards improving plant yields. Okay, so these are the sort of advantages of these PGPMs, right? So stimulate plant growth and productivity, which leads to yield enhancement, biotic and biotic stress tolerance, detoxify soil, biocontrol activities, and modulate the production of phytohormones. So meaning that it provides a better conditions or environment for the plants. Okay, so the effects of biofertilizers containing these different PGPMs or combination of PGPMs on bacterial fungal communities in the rhizosphere soil of different types of crops have not been fully clarified. So people are now looking at the effects of all these biofertilizers. Some of them like applied singly, and some in combination, and then maybe different combination, and people are looking at the effects towards rhizospheric microbes. Okay, so the effects could be in terms of physical and chemical properties of soil, rhizosphere microbiota, of course, plant growth and yield, and perhaps stimulate the native microbiota. So some studies also looking at the effects of decomposition process. So how they affect the patterns of organic material decomposition process. So when it stimulate or it change the native microbiota, so the process of decomposition also might be affected, something like that. Okay, so I have I bring two papers related to this topic in order to discuss with. everyone today. So one of it, the study, the paper is effects of biofertilizers application on soil biodiversity and litter degradation. So the one I've mentioned just now, looking at the decomposition process. So this paper aimed to determine if the supply of biofertilizers could differently stimulate the native microbiota, contributing to different patterns of organic material decomposition process. It's been conducted in an apricot orchard in Italy with two different types of biofertilizers, the Arbusculum mycorrhizal fungi and Trichoderma, were compared to unfertilized control over 1 year. Experiment or the observation also very important, and I realized that normally people do the experiments for about 6 months to over 2 years, like that. So the microbial composition of soil and litter was investigated by using, of course, next generation sequencing, meta-barcoding approach. So the chemical structure of decomposing litter bags was investigated through TGDT analysis. And so this one for the chemical parts. And the results shows that the supply of biofertilizers partially modified the bacterial community. And as we can see that almost after half of year, then only we can see the modification of this bacterial community, okay? And also it influenced fungal community, and somehow Trichoderma managed to cause faster and higher degradation of litter or the organic materials, okay? So the other one, to determine the effects of biofertilizer application on rhizosphere soil, physicochemical indicators, microbial community composition, function and network patterns using, of course, NGS as well, 16S and 18S gene amplification in order to look at the diversity of bacteria and fungus, okay? I'm sort of running out of time. So these are the setting. I will quickly go to the results. So biofertilizer was effective in increasing yield, okay, the sugarcane yield compared to control or compound. This is compound fertilizer, so chemical fertilizer, right? And then also cause a lot of improvement in terms of the soil fertility and productivity, change the diversity of fungi and bacteria as well, and greatly altering composition and structure of the interroot microbial community. And another interesting results mentioned is that based on network analysis, the network structure of biofertilizers was closer to the network characteristics of healthy soils, indicating that biofertilizers can improve soil health. So now people are trying to look at the healthy composition of microbiota in soil, so that we can mimic that kind of composition to other conditions or environment in order to improve the soil health of other areas, something like that. Okay. So quickly perspective, understanding how the rhizosphere microbiota change and how they can be harnessed is a basis for manipulating soil microbiota either directly by amending soil with specific microbes or indirectly, which can be done by altering management practice to promote plant health, right? And then knowledge on rhizosphere microbiome, develop strategies for shaping the rhizosphere to benefit the plants. So using all these omics tools, we can easily understand the plant microbe association, which can somehow draft the future biofertilizers, okay, the composition or the cocktails instead of just applying single or 2 or 3 types of biofertilizers. Okay, so tools and challenges. So can I proceed?
Yeah, sure.
Yeah, and it's a bit weird, like talking to the screen and can't see anything. Okay, so culture dependent methods and culture independent methods is a tools that being used, right? And I believe that these two methods should still be applied together, because for those culture independent, we are able to identify those non-cultivable microorganisms and even the microbiome function, right? Using but then getting the isolates is also very important so that we can focus on the specific microbes that somehow able to be put into that group of biofertilizers, those PGPMs, so that we can somehow use that specific PGPM in order to improve the native microbiota of the soil. So something like that. And then peeking into the ongoing studies of my own study. So I have one project related to this the use of biofertilizer, looking at the effects of trichoderma application on the rhizosphere microbes of rice field. So the objective is to assess the community changes at the rhizosphere of rice. That being studied. So now we want to do it, we want to apply under field conditions and look at the bigger picture, the effects towards other microbes. So we use 16S and ITS metagenomics analysis to unveil the significant effects of this SL2 strain on the selective recruitment of specific microbes in the rhizosphere of rice plants. So this paper being published related to our trichoderma aspergillum SL2, where it's been proven that it can sort of promote plant growth and yield and whatnot. So now we want to look at the effects towards the rhizospheric microbes in the natural condition. Going. This pie chart shows the ASPs assigned to each phylum, the whole results, and then limited data and at the very beginning of the project. So we can conclude that the application of trichoderma is not really causing any significant difference compared to the control environment without the application of trichoderma. So like I mentioned earlier just now, the study should be conducted at a longer period of time, up to 1 year, something like that, in order to see the modification of the microbiota, the soil microbiota, based on the application of the biofertilizers. And these are related publications of these topics, okay? And yeah, so with that, I really like to thank, express my sincere gratitude to all these people here that are collaborating with me in these studies. And thank you for listening.
Right. Thank you, Dr. Nurul Shamsina, for sharing with us these interesting works. And but before we open for Q&A sessions, there's a link for the evaluation form posted in the chat box. So I hope everyone can fill up the form and giving us some feedback on the seminar. So, all right. So if anyone who has any questions would like to ask Dr. Nurul, you may either switch on your mic and ask directly, or you could also post your questions in the chat box.
Dr. Nurul, Fadli here. So anyway, thank you for the talk. I thought it was a very quite in-depth overview of your field. I guess I have two questions. I think it's more on the environmental side of this thing. So number one is you talked earlier about the concern on the overuse of chemical fertilizers, right? And that's why we want to use microbiome as an alternative solution. So is there such thing as the possibility of overuse of microbiomes as well. So that's number one. And secondly is because I come from plant engineering side of research. So in plant engineering, we do have this, we are able to use non-native genes that can be integrated into plant genome to express certain proteins. So I wonder if in your area, like, is there also such thing as the use of non-native microbiomes in the soil, and whether if there is such thing, will it what are the effects of it, and how, if there are any negative effects, how people are addressing this issue? I think those are my questions.
Okay, thank you so much, Dr. Fadli, for the questions. So overuse of microorganisms or
overuse
microbes, right?
Mm-hmm.
Okay, so this is sort of topics that being discussed also among those organic people. We can say that. So organic peoples, they against all the chemicals. And then, to be honest, like myself, I'm still like believe in integrated farming system, where we still use a bit of chemicals, for example, to control disease and whatnot, especially outbreak. And then we apply all these microorganisms so that we enhance the defense system of plants, right? But then overuse of microbes, it's not really significant, I would say, like chemicals, okay, because those microbes actually being isolated from the soil as well, and then they are living organisms, and then they will somehow adapt to the conditions, and then but then, like I mentioned today, like biofertilizer effects towards the native microorganisms, right? And then we can see a shift in the communities, in the composition, because of the application of biofertilizers. So sort of non-native microbes, I think like that these two questions sort of related. So they somehow affects the composition, but then so far people reported that it is in a positive way. So meaning that they promote growth of other beneficial microbes as well, and then they mitigate or control the growth of pathogenic microbes in soil. So, yeah.
Okay. Thank you.
Thank you, Fadli, and thank you, Dr. Nurul. So, is there any other questions? Yes, someone is. Okay, Fikri.
Hi. Assalamualaikum. Good
Hello.
morning, everyone. Right. I
Assalamualaikum.
have a question for Dr. Nurul. I think you have my question is regarding the time it takes for the biofertilizers to take effect. So from what I gather from your presentation, it takes around 6 months or more than that in order to get the intended effect. So how do you see the application of biofertilizers for the usage for the crops that have shorter rotation, like it only takes like 1 month or 2 months before harvest? Yeah.
Okay, thank you for that question. Yeah, when we talk about the effects of biofertilizers, right, we look at the response of farmers. So they use chemicals in order to control disease, in order to promote yield of plants. And then when we talk about beneficial microbes, so they normally don't see the effect so fast, right, during the first season. So nothing happened. And then they sort of frustrated.
I guess,
yeah, right. And then they opt for chemicals again. But then, as we all know that they are living organisms, right? And then they need time to adapt. And they need time to also first adapt, then only they can show their performance, right? Something like us also. So we need to know that the effects is somehow slower compared to chemicals, right? And if you're talking about those short-term crops, it doesn't matter. Of course, you can't see the effects during the first season, second season, but actually you're improving the soil quality in the long term, right? And towards the end, actually, we are also promoting a better environment and especially soil.
I guess I got it wrong. Like I thought it's like a quick remedy, quick solution for better plant growth. Like you start the treatment this round of crop, and then you can get the effect within at the time of harvest. So basically, it's a long run kind of process. Like you actually what do you call it? Improving the health of the soil first, then you can reap the benefits in terms of the crop yield.
Yeah, true.
Yeah.
That people now talking about food for future in 2050, increase in human population and whatnot. So we need to take the steps now in order to have a better future, right?
Yep. And reduce our reliance on chemical fertilizers. Thank you very much, Dr. Nurul. I appreciate the
Thank you.
Yeah, just curious regarding the efficacy of the biofertilizer, right? Just now, when Dr. Fikri and you all discussed. So I was wondering, like, how frequent actually we should apply the biofertilizers in general in the field conditions.
Okay, it depends on crops, I guess, and then normally how people practice if taking rice, for example, because I did research on that. So people the farmers will somehow treat their seeds with the PGPM first. So meaning that they prepare the seeds with defense, and then there's something like applying chemicals as well instead of the chemical fertilizer NPK and whatnot. So they choose to apply microbes based fertilizers.
So for example, in your research, you are going to do on rice, right? So that means you also have to frequently apply it throughout the whole planting times.
In terms of application, actually it's a complicated things as well, because if let's say they are still practicing conventional methods, meaning they still use chemicals, so of course it will affect the population of these biofertilizers in the soil, so it will be depleted and then we need to apply again. But we need to understand that they are living microbes, right? So they can propagate and then they adapt. So the effect is slow, but the presence. And then I believe that now people are looking at the sustainability of the biofertilizers as well. After 1 year, what will happen after 2 years, something like that. So whether they can still sustain, or whether they are being killed by other microbes. So that kind of communication among these microbes is actually still under research, I would say. Yeah, we need to do more research on that instead of just looking at the effects in glasshouse experiment, and then we know that this can promote growth, yield, and whatnot. Then we apply, and then not knowing like how's the effects in long run.
Yeah, I think that will be really interesting to see, what's the effect of applying the biostimulants, and then how this microbe can actually interact with each other, with the populations, as well as to the plant as well.
Yeah.
Right. Is there any other questions?
Can I have a short one? Another one from Fikri.
Sure.
I just want to refer to your phytobiome figure. So you showed that many components contribute to the whole phytobiome, right? And one of it is microbe, but also there's also the component of the climate, which we know we are suffering like the effects of climate change right now. So I wonder how the climate factor would affect the efficacy of the microbiome or the biofertilizers, because they are also, I think they would also what do you call it, would be affected by the climate change, and not necessarily the plant, but the microbes themselves would be affected by the change in the climate. So how can they? So I would see that if you use biofertilizers for a long run, but then you cannot control the climate. So the climate change that would affect the efficacy or the effectiveness of the biofertilizers. What do you think?
Yeah, I think it's sort of personal opinions, maybe not a very concrete answer, right? Yes, of course, microbes will be affected by climate change. But then if you look at the current research, people are trying to find those microbes that salt tolerance, what else, like temperature can sustain or survive in a high temperature, something like that. So perhaps in future, in that cocktails of microbes that we are going to apply, we will include these microbes that somehow can protect other microorganisms in that kind of harsh environment. Yeah, right.
So probably
And then,
like you need to like mostly people with studying biofertilizers would need to like consider like extreme condition and how to plant crops, and then what are the microbes that can be helpful in that situation.
Yeah,
my 2 cents.
And one thing I like about microbes is that we can learn what do you call that, working together, networking from microbes, actually, how they are nothing if they are just one cell like that, but they can cause a huge impact when they are together. So
yeah, the collaboration between
collaboration, right? That's the word.
All right.
Okay.
Thank you.
Thank you so much for all the comments.
Yeah. Is there any other questions from others? Okay. So just a quick reminder. So please fill up the evaluation form for the seminars for our record as well, and also the improvement. So I think I guess there's no questions in the chat as well. So yeah, so I think that's all for today. So thank you very much, Dr. Nurul Shamsina, for sharing her, the research work and some of the knowledge about the phytobiomes. Oh,